Microbe TV. This is Immune, episode number 99, recorded on November 18th, 2025. I'm Vincent Racaniello. You're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, and welcome back. How are you?
I'm all right. I just traveled for a week and I'm back.
You made it back.
It's good to
That's
be back. The body's defenders against disease and also against itself we're going to talk about today.
Yeah.
Also joining us from Madison, New Jersey, Breanne Barker.
Hi, great to be here and great to see you all. I'm super excited to talk about immunology today. I mean, I'm excited to talk about it most days, but...
Most days? There are days you don't want to talk about it?
Maybe if I'm on vacation.
Well, it's winter. It's 9 degrees Celsius and cloudy here in Chelsea.
It's cold and cloudy here.
I was out in LA.
That's from now until
and
May.
It was raining and cold.
Oh.
Yeah.
Yuck.
It's true. It's true. Till May, it's it. If you like these programs where we explain science to you from the original papers, no lies, no disinformation, we'd love your support to continue them. You notice we don't do ads. I don't like ads, especially in educational materials. So we depend on your donations to do these programs. You know, Microbe TV is a nonprofit in the U.S., so if you donate, your contribution is federal U.S. tax deductible. Go to microbe.tv/contribute. A bunch of ways you can help us out. Today, we are going to talk about the recent Nobel Prizes in Physiology or Medicine, which were given to Mary Brunkow, Fred Ramsdell, and Shimon Sakaguchi for finding and understanding regulatory T cells, or T-regs, which are CD4 positive, CD25 positive, and FOXP3 positive. And I love fox because it makes me think of foxes, which are lovely animals. Don't you love foxes?
Sure.
So, as you know, and you know, we've talked a lot here on Immune and on other podcasts, you know, our immune system has a great capacity to recognize foreign antigens, right? We have B cell receptors and T cell receptors that are so variable. It's incredible. But sometimes it goes off and you end up recognizing yourself, leading to like an autoimmune disease. And so that's where this year's Nobel is in. And it's really a lovely story that involves, you know, two separate groups and a great example of collaboration, not collaboration, but working on the same problem and kind of doing independent things.
Yeah, so findings by one group helps fuel the discoveries of the other group, and it goes back and forth. Yeah, I thought that's really nice.
Yeah, no, that's a really great part of this. And this is something that sort of, like many aspects of the field, you know, has this sort of long, convoluted history.
Oh, yeah.
That is kind of interesting to think about, and interesting to think about places where we may be doing similar things today, as a cautionary tale.
When I was a grad student in the 70s, there was a guy at Yale, Richard Gershon, and he published papers on what he called suppressor T cells.
Yep.
Okay, so the idea that certain T cells could suppress immune responses. He published that as early as 1970, but it was kind of messy because the technology back then was not very good, and that went away basically, and there was no more mention for many years.
Yeah, so part of what happened is that there's some sort of genetics work trying to find a gene that was related to suppressor T cells, and many of the researchers came upon this gene called IJ. And IJ, you know, sort of involved in MHC class 2, involved in recognition, I think, of like IG heavy chains. There was this whole IJ story. And then when more careful genetics were done on the MHC locus, it turned out there was no IJ gene.
Just didn't exist.
It didn't exist, and there was nothing that would recognize heavy chains in the way that was described. And so when I was an undergrad in immunology, working in an immunology lab, I kind of learned two things in the lab that were absolute no-no words that you never said, that no, real immunologist who knew what they were talking about would ever use these two terms. And it was sort of like I was told, like, if you ever say these two words, lightning's going to strike you down. And one of those words was suppressor.
Yep.
Yeah.
Anything involved suppressing or suppressor. And the other word was danger.
Interesting.
And so it works out that both of those fields, thinking about danger and sort of suppressors, kind of revitalized really early in my time as a graduate student. And it still strikes me as really funny how much I was taught those were no-nos, and how now they're such major things in the field. And so, there was sort of this problem with IJ. IJ kind of crashed and burned. And then it was sort of like there was this time where a lot of immunologists were like, what? Suppressor T cells? I never believed in that.
Yeah.
What are you talking about? That's
they
not
can't exist.
They can't exist. And I never believed that. I don't know why you say I did, but I never believed that. That was not a thing. And so again, I think that this is really interesting to think about and just to remember as we are looking at different areas of the field to wonder about, in the future, are there things that we currently think that our technology is going to give us a new appreciation of later on?
Maybe things that are correct bubble to the top eventually.
Yeah.
Yeah.
So I'm at Columbia, and Richard Gershon's brother, Michael Gershon, is at Columbia. And
I didn't know that.
He's a professor in cell biology.
Mm-hmm.
And he always said, my brother was right.
Yeah.
I mean, the details may not be, but in the end, this idea of suppression or regulatory T cells came back.
Yeah. And there were people working on that. And their experiments, had holes in this whole IJ story and things like that. But the Nobel Committee did not recognize that body of work that set the foundation for the existence of these cells.
The other thing to mention is that when sort of we had this renaissance of the field, and the body of work that sort of did start us on the track that we're going to talk about here, the word suppressor still kind of went away.
We rebranded them.
Yeah, there was a rebrand, exactly as Cindy said. And so now immunologists often use the word regulate when they want to say suppress, but they feel like they can't, or turn off. And so these are the regulatory T cells. And so I think that that's also an important thing to note, is that that history is part of the reasoning for the terminology we use today.
Yeah.
Unfortunately, Richard Gershon died at 50, and he never got to see any of this, right? Would have been...
that's so disappointing.
It would have been nice.
Yeah.
But
he could have said, I told you so.
Just like, the U.S. switched back to Salk vaccine in 2000, and Salk wasn't around anymore, but he would have loved to see that. Many examples of that.
Yeah.
So we're going to do a paper, but just a little bit more background. So this whole field is revitalized, in the 80s and 90s. Mm-hmm. And there are experiments with in Japan by Yasuaki Nishizuka and Teruo Sakakura. They showed if you take the thymus out of newborn mice, they get autoimmune diseases, right? And another group showed the same thing in rats. And so, as you might guess, Nishizuka and Sakakura are working in Japan. And then Shimon Sakaguchi decides to join the Nishizuka lab because he's interested in this. And he finds that taking the thymus out of rats, you could get around that by adding back a specific fraction of immune cells, which were characterized by specific markers.
And talk about terrible immune terminology. Those antibodies were originally called anti-LYT1, which sees CD5. I'm not sure I even still know what CD5 does, but it's on activated T cells. It marks T cells. Yes. But yeah, it's hard when you go back in the literature sometimes to figure out what they're talking about, because the nomenclature changes over time, and these antibodies got redefined. They were originally made to different markers and they got different names, but then when we started classifying them as clusters of differentiation, or CD, then they all got these CD nomenclature. And so you go back in the literature and sometimes it's hard to figure out what the heck they're talking about.
And Sakaguchi ends up doing some studies that are in a paper from 1995 that I really think of as sort of the beginning of a lot of this. Yes. It's sort of the paper that starts a lot of aspects of this field. And it has a lot of really good parts, but it also has this one kind of challenge that comes with it.
Yeah. So that paper, I think you're talking about the one that was in Journal of Immunology.
Yes.
Yeah. So if you like immunology and you follow this kind of thing, occasionally they do these things called pillars in immunology. And this was one of those pillars papers. So they pull out some fundamental paper that defined the field or defined a concept in immunology. And so they years ago kind of had this idea that was really important. So they preceded the Nobel Prize for this, but they did highlight this particular paper, and there's a nice preview that goes along with it too, that describes the history of the field.
Yeah, I highly recommend the Pillars series and those Pillars sort of history articles to anybody who's really interested in the field. This one was from April 2011, where they republished it and published about the history. And I found it so interesting to read at the time, but I also, this paper we taught in grad school for a long time because it is so key and so revolutionary.
Yeah, it's in the Nobel description. They call it a landmark study.
Yep.
So Sakaguchi had found that some subpopulation of CD4 cells, CD4 T cells, were involved in regulatory activities. And here in this paper, he shows that they are CD4 T lymphocytes expressing CD25.
That's right.
Right. And in fact, if you add CD4 positive, CD25 positive T cells into these thymectomized animals, it prevents autoimmunity from happening. So suggesting those cells are important for this, for regulating autoimmunity.
Yeah. And so that was kind of the first idea of the T-reg, was that it was a CD4 positive, CD25 positive T cell, and goes back to this awesome paper from Sakaguchi. But there was always a little bit of an issue here. And the issue is related to CD25 and the identity of CD25
because I might not know what CD5 does, but I do know what CD25 does.
So CD25 is also known as the IL-2 receptor alpha. So it's one of three chains that's involved in the IL-2 receptor on T cells. And big picture, this actually might be important for regulatory T cells because there's some idea that, in fact, they might be sort of IL-2 hogs because they have this high affinity.
Soaking up all the IL-2.
Yeah, soaking up all the IL-2 because they have this high affinity IL-2 receptor chain, CD25. The reason why this is a problem is that CD25 can be on T cells for other reasons. So there are other situations where CD25 is on T cells. Specifically, when a T cell is activated, sort of recently upon its stimulation, recently upon its activation, it turns on CD25 in order to get a lot of IL-2 signal. And so recently activated T cells are CD25 positive. And so one of the issues in the field for quite a while was, okay, there seem to be these regulatory CD4 positive, CD25 positive T cells, but there are also other CD4 positive, CD25 T cells that are recently activated cells. And in some ways, they're almost the exact opposite of regulatory T cells. They're the really active ones that are causing damage. And so,
if you deplete them, what would happen?
Yeah. And if you deplete them, then you are going to, change phenotype as well. And so one of the problems here, so this paper is a fabulous landmark study, but CD25 wasn't that specific of a marker. Mm-hmm. And could mark some other cells with another really important phenotype. And so I think the field, at least my impression, and again, this gets into sort of timing of where I was in different parts of my training. I think that, people were like, okay, this is sort of good. This kind of makes sense. But without a really specific protein that was unique to these cells and not to other cell types, it was really hard to move any of it forward or to really sort of solidify any of these questions.
And that marker would be most easy to use and exploit and do experiments with if it were on the surface. Right. That's why Sakaguchi was doing all of this, to try and look for a surface marker, because that makes it really easy to pull out that population or add it back or dose it in at different concentrations or use lots of different models to try and address that. If you don't have a surface marker, it becomes really, really difficult.
Yeah. When I read that Sakaguchi paper, that original Sakaguchi paper, I often kind of wonder about some of the background, because it's sort of like they do some things with CD5, as you mentioned, and then they end up with CD25. And I'm kind of like, did you go one by one? Did you go try each number until you found one that kind of worked? Because there are times where it feels like that might have been what they did.
Sometimes they have brute force and go through several trainees before they get to the right molecule.
Well, that's the second part of the Nobel Prize. This is now Mary Brunkow and Fred Ramsdell. They're working at a company in Washington called Celltech Chiroscience. And they're working on this mouse called the scurfy mouse, which develops autoimmune disease. And so they just sequenced the genome of this mouse. It's amazing that they were able to do this. And they're able to narrow down the responsible mutation to the X chromosome. And they find a 2 base pair insertion in a gene that makes a stop codon that seems to correlate with autoimmune disease. And they call the gene forkhead box P3 or FOXP3. So now we have a candidate marker for something more specific than CD25.
The sad part, of course, is that a forkhead box protein is a transcription factor.
Yes,
so it's not going to be on the
surface.
Right.
Right.
Yeah, it's too bad. But sometimes you have to look elsewhere. You can't just look where the light is.
Yeah. But it's important to know this, because if you want to try and this becomes really important for the paper we're going to talk about, if you want to try and look at function, and you have to destroy the cell. Basically, you have to fix the cell to be able to see if it expresses the gene of interest. How do you do functional studies? Like how if you know CD25 is a marker, you can pluck it out and see what happens. You can put it back. The cells that have it, you can see what happens. But and you can also pull them out and look at their function. So you can see if they have activity in vitro. If you have something that's inside the cell, in order to see if the cell expresses it, you either have to lyse it and look at the RNA, or lyse it, look at the protein, or fix it and stain cells inside. But once you do any of one of those things, you can no longer analyze its function. You can't add it back. You can't do all of these things. So inherently, it's much easier to look for something on the surface, because you can then test its function. Whereas if you find a transcription factor, it's now exponentially more difficult to try and figure out whether that's actually responsible for it or not. And it plays into, what they try, how they approach that question with this paper.
So what they do is they make transgenic mice that produce FOXP3. They cross it with the scurfy. Now the scurfy doesn't develop autoimmune disease anymore. So they publish that. And of course, Sakaguchi hears about it. And what is he going to do? He starts to work on it.
Yep.
And that's the that's the paper we're going to look at today.
One other thing that Brunkow and Ramsdell did is they were sort of looking at this FOXP3 gene, and they found that there were humans who had mutations in this gene. And those individuals, usually little boys, because it's an X-linked gene, have a disease called IPEX, which is immune polyendocrinopathy X-linked syndrome. Aren't you glad that this was in my lecture yesterday?
I was like, I gotta look that back up because I can't remember what that stands for.
And in IPEX, these kids have a lot of autoimmune symptoms against a number of different body systems. So they have, in particular, some against endocrine glands, but they also have a number of other kind of problems, and most of them will not make it through the first few years of life. So it's a pretty severe disease. And it's basically a defect in, making central tolerance work in these individuals. And in fact, they also have this mutation in FOXP3. So that sort of backed up their data on the scurfy mouse. All of this are happening around the same time.
And I will add, so Shimon Sakaguchi must have been reading outside of his area, right, to come across that paper. And I think the key piece here is that the scurfy mice, the IPEX humans, and his model system, in which the prior model system where you thymectomize the mice, or the model system that he was using where if you deplete the CD25 positive cells and put the remaining T cells into a mouse, it causes autoimmune disease. So all of those things together looked similar in the type of pathology that they caused, although it's still very general because they cause autoimmunity, and some cause more endocrine problems, some cause more colytic problems, but generally they would all induce a similar type of function. So I think that's why he was like, I wonder if that might be the key gene that we're looking for to define what a T regulatory cell is.
So this paper was published February 2003 in Science. It's Control of Regulatory T Cell Development by the Transcription Factor FOXP3. The authors are Hori, Nomura, and Sakaguchi. And the abstract is beautiful. I want to read it because it's so clear. So there's some abstracts today are so hard to read. You have to read them over and over. But this one, look, regulatory T cells engage in maintenance of immunological self-tolerance by actively suppressing self-reactive lymphocytes. Little is known, however, about the molecular mechanism of their development. Here we show that FOXP3, which encodes a transcription factor that is genetically defective in an autoimmune and inflammatory syndrome in humans and mice, which we just talked about, is specifically expressed in naturally arising CD4 positive regulatory T cells. Furthermore, retroviral gene transfer of FOXP3 converts naive T cells towards a regulatory T cell phenotype similar to that of naturally occurring CD4 positive regulatory T cells. Thus, FOXP3 is a key regulatory gene for the development of regulatory T cells. It's beautiful, right?
Yeah. I guess one other thing that will come back a little bit at the end of this paper is there was also a sort of development around the same time of a type of cell called the Th3 cell that seemed to be cells that could become regulatory T cells, particularly in the gut, in sort of they could be induced to become regulatory T cells. And so there was already this idea from Sakaguchi that there was something about some regulatory T cells coming out of the thymus and sort of being born as regulatory T cells, and some regulatory T cells developing or getting polarized into regulatory T cells later. And that does come in at the end of this paper. So he talks about things like the naturally occurring and the naturally arising regulatory T cells here.
So the background in this paper, we just discussed all the background. He's talking about scurfy mice. And interestingly, FOXP3 was also called scurfin.
Yep.
Which I like. I like FOXP3 better. I like foxes better than scurf. But
I always was pronouncing it wrong, like scruffy mice versus scurfy. What is scurfy anyway?
I don't know what scurfy is.
It seems like a weird name.
Scruffy, I understand.
Scruffy.
Right?
Yeah.
You could see how an autoimmune disease might make a mouse scruffy, right?
Yeah, sure. But scurfy? I don't know.
So the first thing they do is look at where FOXP3 mRNA is expressed in the thymus.
Sorry.
What's up? Yeah.
I Googled what does the word scurfy mean.
Oh, what did you find out?
What does it mean?
It means having or resembling scurf, which is a flaky, scaly, or crusty outer layer on the surface, like dandruff, scales on a plant, or flaky skin.
Okay, so then that's probably what the mice look like. So it's fair enough.
Makes
fair
sense.
enough.
Yeah, it's good.
Enough. Yeah.
So they do RT-PCR to look at FOXP3 expression.
Standard PCR. Old school.
Just standard. Oh, yeah.
Old school.
Standard.
Old school PCR.
Yeah, you got a machine and it just cycles.
You got a band or you don't have a band.
Yeah, they run gels. That's right. So they find that in the thymus, so the CD25, CD4, CD8 negative, CD25 positive, 4 positive, CD8 negative thymocytes, that's about 5%, predominantly make FOXP3 mRNA. But the CD4 minus CD8 positive populations do not. That's in the thymus. And in the periphery, CD4 positive T cells also transcribe the gene, but CD8 positive and CD19 positive B cells do not. Why did they look at B cells? Just as a control, right?
Just as a control, I would think. Yeah.
Okay.
They wouldn't. I don't know. Maybe they would express. I don't know if B regulatory cells express it. There are B regulatory cells now too.
Yeah. Right.
But I'm not. I think FOXP3 is specific to CD4 regulatory T cells.
And then they do some real time, Cindy. I'm sorry. They did do a little more advanced PCR.
They did.
So look at levels, and they find that the levels of FOXP3 are 100-fold more abundant in CD25 positive versus CD25 negative CD4 cells. So they can.
Yeah.
Go ahead.
Oh, I was going to say it's also they look at this thing called CD45RB, and they have high and low.
Right.
And it's interesting because one of the populations that Sakaguchi was looking at in that JI Journal of Immunology paper we were talking about, where he found CD25, was they were originally characterized by CD45RB low. So they do have higher expression of FOXP3, so it kind of fits with his original model as well.
Okay. They also find that the expression of FOXP3 doesn't just happen when T cells are activated, because they can do in vitro stimulation of CD25 negative CD4 cells with monoclonals to CD3, and they don't get FOXP3 expression.
Yeah, so this isn't like CD25 and that it just comes on during activation and goes back off.
Right.
So then they ask whether if you forced expression of FOXP3 in naive T cells, whether it would make them regulatory T cell-like. So they basically use retroviral vectors, which encode either green fluorescent protein or green fluorescent protein and FOXP3. Okay, and they use an internal ribosome entry site to get two proteins made from one mRNA. I was working, and these were discovered in picornaviruses, right? So I was working at the time when these were discovered, these irises, and then people immediately picked them up for research use because you could make two proteins from one mRNA. This is very cool.
And that way it was really the GFP level pretty accurately tracked the amount of the FOXP3 protein that you were making. Because remember, we still don't have an antibody for it. And if we did, we'd have to fix the cells, permeabilize, and stain for it. So this provided you with a way to visualize those cells that were making FOXP3 without having to damage them, so that you could then study them functionally and then look at them potentially in vivo later.
Okay, so, and there you go. You got iris, and then you have GFP, both of which are accidental discoveries.
Yep.
And just further emphasizing the serendipity of science, that I wish people would understand more, that you got to let scientists just be curious, right? And good things will happen. And so many people don't get that, right? I mean, GFP came from jellyfish.
It did.
No, why should we study jellyfish, They don't do anything. Okay, anyway, so they take peripheral CD25 negative CD4 cells from normal mice, they stimulate them with antibody to CD3 and IL-2, and then they infect them with either of these two retroviruses. And a week later, they look at proliferation to after T cell receptor stimulation, they look at cytokine production and cell surface molecules. So this infection with the retrovirus, you get good GFP expression, 30 to 60% of the CD4 positive cells. And then after TCR stimulation, the GFP positive cells from the FOXP3 infected cultures grow poorly. They divide poorly in contrast to just the GFP positive without the FOXP3. They proliferate madly. Okay. So that's a reflection of suppression, right? Or I'm sorry, regulation. I used the suppression word. Sorry. And these FOXP3 GFP positive cells make very little IL-2, interferon gamma, IL-4, and IL-10 compared to the control cells, which make a lot
Can
of those,
I just ask one clarifying question? I thought that in this study, they add fresh T cells, which would normally respond, and then they're dosing in either the control or the FOXP3 cells.
Right.
Did you understand that differently?
Let me double check.
Because I think it's CD3 stimulated response of freshly isolated CD25 negative CD4 positive T cells, plus then either the FOXP3 expressors or the controls. Because that way you're looking actually right away at the suppressive activity of those cells, which is, I think it's pretty impressive that this actually works. Like they're able to take some of those cells and make them into regulatory T cells just by expressing FOXP3.
Well, they do that for the proliferation, but they don't think that's what they do for the cytokine.
Oh, okay. So
I think
just
the cytokine, I think they just do that for the cytokine, or for the proliferation. Sorry, because one of the things that sort of stands out when you look at this paper now is where's the IL-10?
Yeah.
And it's sort of interesting that they're not seeing IL-10 here.
Nope.
Which, at least according to the textbooks now, would be one of the things that these cells would be making.
Right.
But again, there's some things that they have at the end of this paper that I think are sort of interesting.
Yep.
And so the basic thing that they sort of mention at the end of this paper is that, because these cells are becoming FOXP3 positive via overexpression, they may have some slight differences between them and sort of a traditional regulatory T cell.
Okay, so they also look at a number of other markers that are on these T-reg cells and basically conclude that if you put FOXP3 into T cells, it can render them hypo-responsive to TCR stimulation, right? Hypo, less responsive. Mm-hmm. It inhibits cytokine production, and it turns up surface expression of molecules that are associated with regulatory function of these naturally occurring T-reg cells. Then they ask, can these FOXP3 transduced T cells have suppressive? Oh, they use this S word. They said suppressive activity. Wow.
Do you see how many times lightning has not struck us down?
But I think we usually say suppressive activity now. I think it's okay. It's not as dangerous to use that word anymore.
So they have, their CD25 minus CD4 positive T cells, which have been infected to produce FOXP3. They can reduce proliferation of fresh CD25 minus CD4 positive T cells when stimulated with an antibody to CD3.
Yep.
Okay. And that suppression they can see by inhibition of IL-2 production in a way that naturally occurring CD25, CD4 T-reg cells do.
Yep.
So
it's definitely sufficient to make what appears to be a regulatory T cell.
Yeah, out of a non-regulatory T cell. Mm-hmm. And they find that these, so if you take CD25 positive, CD4 positive cells, not before putting in FOXP3, those are equally suppressive with or without FOXP3 because they already have it turned on, presumably, right?
Yep.
Okay, then they ask whether antigen-specific T cells can be converted to regular T cells by FOXP3 expression, right? So they have CD4 positive T cells that are specific for ovalbumin peptide, right, which have previously been made. So they have a TCR that recognizes that. And they barely have any FOXP3 to begin with, right?
Right.
So they take, these are transgenic mice, right? Mm-hmm. So they take them, they take the CD4 positive T cells and they affect them with their FOXP3 construct. And they find that this suppresses the proliferation of fresh, non-infected transgenic T cells when they add OVA peptides, right? Because that's going to be recognized by these T cells. And if you have FOXP3 in your other CD4s, it's going to suppress that. So it shows that ectopic expression, which just means you've added FOXP3 in, is enough to convert non-regulatory T cells towards a regulatory phenotype, which can suppress proliferation of other T cells, which is through inhibiting IL-2 production.
Pretty cool.
And these T cells look like they're suppressing in a similar way as do CD25, CD4 T cells, right? They looked at a bunch of parameters and they look very different. And they say there are three reasons why this is so. First, these FOXP3 cells fail to suppress these CD4 cells from the transgenic mice when stimulated with OVA, but polyclonal stimulation with CD3 antibody induces suppression. So in other words, the FOXP3-induced T cells require stimulation through the T cell receptor to exert suppression. They have to be touching each other, right? Second, these FOXP3-infected T cells do not suppress other T cells when they're separated by a semi-permeable membrane, right? They have to touch each other. Third, neutralization of TGF-beta or blocking IL-10 receptor failed to block suppression. So in vitro suppression by these FOXP3 T cells is not probably through soluble factors, but cell-to-cell contact. So what would be the last experiment you would do? How about disease? Could we look at disease?
Because wouldn't it be cool if you could generate regulatory cells and treat a disease that's like an autoimmune disease, and if you put back some regulatory cells, you could treat that. That would be cool.
Right. So they use a model of inflammatory bowel disease and autoimmune gastritis, which can be induced in skid mice by transferring these CD25 negative, CD45 RB high CD4 T cells from biopsy mice and prevented by co-transfer of CD25 positive T cells. So they have CD25 negative, CD45 RB high, CD4 from biopsy mice. They're infected with their retrovirus constructs producing FOXP3 or the control. They are co-transferred with freshly made CD25 negative, RB high, etc. cells. And these FOXP3 cells that they have made to transduced ones inhibit weight loss, diarrhea, and histological development of colitis and gastritis induced by the transfer of CD25 negative cells into those mice, as well as normal CD25 CD4 cells do. And of course, if you put just the GFP control in, no effect on disease. Right. So it works.
And it's very clear too. I mean, it's like all the mice get disease, all the mice do not get disease.
Yeah, you don't need any statistics.
No. Well, they didn't do any either. Well, they did on some of them, but not on the colitis scores and gastritis scores.
So those are the experiments.
And if you like this stuff, go look at the histology images, because you do not have to be a pathologist to see the differences. No. It's pretty clear.
Yeah. So FOXP3 is expressed in these CD25, CD4 cells naturally. And if you put FOXP3 in naive T cells, it can convert them to T-regs. They also point out that, I think you mentioned this before, right? The mutations in FOXP3 gene in humans give a multi-organ autoimmune disease, both in mice and in people. Oh, and another thing in this paper, which they did, which is in the supplemental figures, they removed the fork head domain from FOXP3, which is essential for its transcription factor activity, and that does not make a T-reg cell. So obviously it's the transcription factor activity of FOXP3 that's doing this, and obviously it's turning on other genes to make a T-reg phenotype. Do we know today what some of those are? I guess we do, right?
We know some about what those are, but there are also a bunch of places where things got a little muddy after this paper in terms of is cell contact required or not? Are certain cytokines involved or not? And I would have put money on it being at the very end of this paper, and clearly I'm not seeing it right now. But there's sort of this question of, well, is some part of that T-regs that come out of the thymus versus T-regs that are made in the periphery? And is that why we're seeing slightly different things in different studies? There's another study from Brunkow and Ramsdell around the same time that has a little bit of a difference in some of that. And I think that was sort of this idea of, are these different populations of T-regs distinct? And some of those I think are still open questions even now.
But I think it raises that
So while
point.
this is happening, Alex Rudensky.
Sasha, yeah.
It's okay, go.
No, go ahead. Talk about Sasha.
Yeah, Sasha Rudensky.
Yeah, Sasha knocked out FOXP3 and showed the mice develop autoimmune disease, which is a nice closure of that circle,
Absolutely.
Yeah, that was when I was a first year grad student. Yeah. He came and gave a talk on those data during my first year of grad school. And so I heard that talk real early, and I remember having no idea what was going on. And now I'm sort of like, that was a really important talk. I wish I remembered it better and knew what was going on so much better than I did.
I bet he didn't explain FOXP3 at the begining. He probably just said FOXP3 is important for T-regs. That's all, and didn't give you any nice background, which would have helped, because he was like that.
I would agree with that, because he talked here and he was giving a really wide university lecture, and his first slide was on the importance of T regulatory cells, and he went deeper and deeper and deeper. And I felt so bad because there were so many people who came from disciplines really far and wide that don't even know what a T cell is, let alone a regulatory T cell.
I'm still blaming my first year studentness.
Well, it just helps if you can, communicate science in a way that at least puts it in context first, right? Because we got into some details here, but at least if you understand, what's happening and how these cells are functioning, it's important.
I mean, it's basic teaching, right? Which a lot of scientists don't want to teach; they just want to tell you what they did, right?
Yeah, they do.
And I've seen him many times, and I always got confused because he doesn't explain, but not just him, other people too.
Yeah.
So the last paragraph of this paper is interesting. I don't know what has happened, but they say this could be therapeutically useful, right? Maybe we could put FOXP3 into T cells and give them to people. So has that ever been done?
I'm not aware of human clinical
I don't
work
think
on that.
So.
But why wouldn't you do it though, right? Is there a problem with it? Like you could take T cells out of people and transduce them and put them back in. But I guess you'd have to keep doing it over and over again.
Well, and would they have to be specific to the antigen or not? And I think that's another, in some cases, point of debate. And if they do have to be specific to the same antigen, then how the heck do you find them to transduce?
Right.
Okay. So you would have to know the antigen.
Maybe.
Yeah. Probably.
Well, you could just take bulk T cells and hope you get a good representation, I guess.
If you took bulk T cells and made them into T-regs, you're potentially making T-regs that will suppress your response to important pathogens.
Yeah.
Right.
That would not be good.
Yeah.
So I think Brianne's point where you'd have to be able to identify those that are specific for an autoantigen for which somebody is making a response, and then take those, expand them, and then make them into T-regs and then put them back, which we also still don't do. Like really much virus transfection or transduction and putting cells back into humans. It's done rarely, but not on a regular basis. Yeah.
So do we know the target antigen for any autoimmune disease?
For many, yes, but not all. I mean, like multiple sclerosis. Lupus has it's more antibody mediated, but we've got antibodies to cardiolipins, to nucleic acids.
There was the lupus paper this week
By the
about
way,
lupus.
That is on my list for the new year that I want to talk about that paper. So if you're listening to
we're
this,
doing it tomorrow.
It's going to come. Oh, you are? Okay, well then you're going to beat us to it.
It's the EBV one.
Yeah,
yes,
the lupus EBV one. Yep.
You're going to be there, Brianne.
I am.
Okay. So,
I don't think it's tomorrow though.
No,
Friday.
tomorrow is Wednesday, right?
Okay, I was like, wow, my
Friday.
week's going fast.
No, yeah, it's Friday. I just put it up early. I rarely put the papers up on Monday, and I did yesterday. And the other paper, in case you're interested, is Watson and Crick, 1953, because he just died. So I thought we'd do his paper, which there's no data in the paper.
It's crazy, right?
It's totally crazy. They do say we're going to publish all the data later, but I don't think they ever did.
Is that right?
I have to check on that before Friday. Yeah. But anyway, so that's this is one of the key papers, I think, in this whole Nobel Prize for this year. And for me, it makes T-regs much clearer.
Yes.
Okay, as a non-immunologist, in particular, reading the Nobel description.
It was really nice, right? That description was really nice.
Yeah, it's really good.
Yeah. So I hope you like that.
I wanted to add one more thing that I thought was important, that, immunologists are kind of clever. It's like when you're faced with a problem and you have to try and figure something out. And it's coming back to this idea of having a surface marker, but FOXP3 is not a surface marker. And it's not great to have to transduce cells all the time to try and figure all this out. So one thing that's been done now is they can lineage trace cells that express FOXP3. There's a couple of different ways to do this. You could knock GFP into the FOXP3 locus. So anytime a cell makes FOXP3, it'll also make GFP. There's some other tricks that people use, but basically what that does is enable you to detect the cells that are making FOXP3 without having to damage them, without having to lyse them, without having to fix and stain them. And so now you can pull out and isolate those cells that have FOXP3, assay their function in vitro without manipulating them, and put them back in vivo without manipulating them. And I think that, those additional studies that followed up on these studies absolutely close that loop to show that, yes, in fact, these deregulatory cells without manipulation, without retroviral transduction or anything else, did actually have this functional capacity, which I think was a really important piece to this whole story. Because one could still argue, although it's controlled for, because you have a control virus that you're transducing with versus a FOXP3 expressing virus, but you still could be doing something funny to the cells. But that way,
so how are they marked? How are they marked? So
it's lineage tracing. So once a cell turns on FOXP3, it'll turn on, either it'll become fluorescent by various different ways. So you could an example is you could put a CRE, recombinase, downstream of a FOXP3 promoter. So anytime the FOXP3 promoter goes on and the cell's making FOXP3, you can now express CRE. And you could, for example, there's a complicated system where there's a stop flox. So there's a stop codon that once the CRE enzyme is expressed, it chops out the stop codon and that gene is expressed. So if that gene is a tomato or a GFP, then any cell that ever made FOXP3 in their lifetime will now become green.
Yeah, even if they stop making FOXP3,
even if they stop making
they will
FOXP3.
Still be green. And so you can sort of trace them forever.
You can sort those cells based on the fluorescence then. Yeah.
That's right. Yep.
Technology.
Yep.
I mean, a lot of technology, fluorescent proteins, Kree-Lox.
They weren't specifically studied for that particular reason.
No.
Right? The Kree-Lox system was just this fortuitous discovery, and people took advantage of that. They found the sequences that the enzyme cut. They started putting them into the genome of certain mice so that you can turn things on when you want to turn them on, so they're not on during development. You can put them on through development and then turn them back off. I think didn't we do the ninja mice where they were flipping antibody sequence? I mean, there's just so many different ways that you can use those kinds of tools to ask very specific questions that would be impossible to ask any other way. It's just so cool.
Yay for
PCR
people.
too, right?
And,
PCR.
And like CRISPR, Kree-Lox came from bacteria.
Yep, that's right. Yeah.
So, I mean, that's why it's all about curiosity.
Yep.
Just, again, and Kree-Lox, cream cheese and lox. I have 2 bagel stores right below the incubator now.
Can you smell the bagels when they're cooking them? That's good, because I think I'd get hungry all the time if I had that.
No, I don't. But if you walk by, you can smell them, yeah.
Oh, yeah. I ate at that when I was there.
That's right. You did.
Yep.
All right. And you're going to be here. Well, when this is released, you will already have been here. Right?
Yep. We're going to all have been there.
Yes. For Immune 100, which when you hear this will have been history.
Yep. So you'd have to wait. You'll have to wait.
Yeah, right. You'll have to wait to hear 100 because this is 99.
That's right.
All right. That's Immune 99. How long did it take? It's 5 years?
No, we started in 2018. 7 years.
7 years.
Right.
Everything happens slower in immunology, doesn't it?
Sometimes, yeah. Because we have to wait for memory cells.
we left out the elephant in the room is that Steph is not here because her son is sick. So that's why it takes us longer too, because, we all have
hopefully
outside
Steph
things.
will be here in December with all of us.
She should be.
And our 2 guests.
We're all planning on being here.
Valerie has just gone out now to look for chairs to rent. Because I don't have 25 chairs.
No.
But we'll find them. You can rent anything in New York. That's Immune episode number 99. If you have any questions or comments, you can send them to immune@microbe.tv. And if you enjoy these programs, you should think about supporting us so that we can make them. These aren't made in my garage, I hire people to help now because I couldn't do it myself after a certain point. And we wanted to grow. Well, like any entity, you want to grow and do more. So we grew and we have expenses. So we need your support to continue. You can go to microbe.tv/contribute. Cindy Leifer is at Cornell University. Cindy Leifer on Blue Sky. Thank you, Cindy.
Thank you. Thanks for the discussion. This was fun.
Brianne Barker is at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Oh, I'm Vincent Racaniello. You can find me at Microbe TV, and I learn more than anybody here because I'm not an immunologist. But I do love immunology. It may be my second favorite subject next to viruses because that's one of the reasons it's there, right?
Yep. They're intricately attached to each other.
Yeah, they're very much intertwined. Although I got to say, everything else, all our other podcasts, I really like them as well. We did a neuroscience yesterday on the neuroscience of overeating. Oh my gosh. I got so hungry. Foods that have high fat and high salt are impossible to resist. Mm-hmm. And that is the basis of a lot of overeating. Anyway, that's going to come out this week, so you can check that out. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.